Hey guys, or Dustin or Chua. My name's Timur. Our group today will be presenting on the HDoc server for integrated protein-protein docking by Yuming Yan. The members of my group are Maddie, me, and Kristina. So just first, I want to give you a little overview of what we're covering today. We're going to start with a just quick introduction to HDoc, including its purpose, flexibility, and some of the challenges it addresses. Then we'll walk through the figures and data in the paper. We'll look at figure one, which outlines the overall workflow of the server. After that, we'll go through figures two to four, which will show the user interface, submission steps, and how the results are displayed. We'll also cover over performance valuation shown in figure in table 5, where HDoc is benchmarked against other docking tools. And then we'll show table 6 and 7, which will go over the server's input quality checks. And then at the end, we'll summarize the key features with a summary and then end with our conclusion and a reflection question. So, the purpose of HDoc is to predict protein-protein and protein nucleic acid interactions. And what makes it unique is that its input flexibility. It can take either 3D protein structures from the PDB or just amino acid sequences in FASTA format. And from there, it uses homology modeling to build structures, even when there's not much structural data available. Makes HDoc really useful for researchers who only have like sequence data, which is pretty common. There are some problems it also helps solve. First, the number of protein-protein complex structures being experimentally determined is growing really quickly. And with that comes a lot of binding interface data like which residues are actually contacting, and incorporating that data into docking workflows can be pretty challenging. Second, in many cases, scientists only have access to amino acid sequences and no 3D structure at all. Most docking programs require structure input, so tools like HDoc that support sequence input are essential for filling that gap. And we'll show how HDoc handles these scenarios as we walk through the figures, starting with the overall workflow in figure one, which we'll be going to. Um, so let's for a brief interview of the workflow. Figure one, I'm going to be talking about illustrates the HDoc server procedure. As you can see, panel A right here outlines the HDoc server's workflow and divides it into four main stages: job input, template-based modeling and structure prediction, molecular docking, the results delivery. So for job input, users submit their data, like protein sequences or structures, and optionally specify binding site residues or distant restraints, represented by the green boxes. And then for the template-based modeling and structure prediction, the server searches for homologous templates in the protein data bank, the PDB, to model the structures of the input proteins. And then for molecular docking, the server utilizes a hybrid strategy and that combines template-based and template-free approaches to predict the interaction between the model proteins. And then for results delivery, the server provides docking results and includes predicted complex structures and associated scores to the user. So panel B illustrates the priority order of the three-dimensional structures used by the HDoc server for docking. The hybrid docking mode is indicated by the red solid arrows and priorities user provided structures. Um, 1 second. Followed by structures modeled based on the homologous templates. And lastly, ab initio predicted structures if templates are unavailable. The template-free docking mode is represented by the orange dash arrows and primarily relies on ab initio predicted structures when user provided structures or homologous templates are not available.
So now we're going to be talking about figures 2 through 4, um, which highlight the user interface and the submission workflow. So to utilize this software, all a researcher really needs is a computer, the amino acid sequence in FASTA format, or structures in PDB format of the receptor and ligand proteins to be docked. And they can optionally have experimental data, such as residue restraints or protein-protein binding. Okay, so now we're going to be looking at figures 2 through 4, which highlight the user interface of HDoc and the submission workflow. So to utilize this software, all the researcher really needs is a computer, the amino acid sequence in FASTA format, or the structures in PDB format of the receptor and ligand proteins to be docked. And they can optionally have experimental data, such as residue restraints for protein-protein binding or SAX data for the complex. They will also need a common web browser like Chrome, Firefox, etc., and an optional molecular visualizing software like Pymol. So to get in a little bit deeper, figure 2 shows the home screen of the HDoc server where users can provide job input for the receptor molecule, ligand molecule, some advanced options, and an optional email address and job name for each respective section. Once the user has provided the job input, all they really need to do is click the submit button and the job will commence. Once the job is submitted, the web interface will be redirected to a page showing the job name and running status that will update every 10 seconds. Users can close the job status page, and when their job is done, they'll get an email notification with a link to their docking results. To talk a little bit more about the optional input data, figure 3 shows an expanded interface for providing the residues of the binding site, one of the more advanced options shown on the home screen. Using these data while inputting, will aid HDoc in guiding the docking process, but it's not required. Figure 4 is the HDoc results after docking has been completed. This will automatically appear on the job status page, or it can be viewed by clicking the link in the email notification. The top of the page is a title with the job name and files for download. Optional buttons on the right can control the viewer, and a docking summary of the top 10 models is shown in a table at the bottom of the figure. And the structure is interactive, and users can use their mouse to rotate the displayed structure. So now we'll get into figure and table 5. They both provide a performance evaluation of HDoc compared to other docking softwares. Table 5 presents a comparative analysis of various docking servers. It demonstrates the HDoc uniquely supports both protein-protein and protein nucleic acid docking, accepts amino acid sequences as inputs and not just structures, and integrates template-based modeling with free docking capabilities. Figure 5 showcases the benchmark performance of the HDoc server in CASP13 Capri blind docking experiment. And it also highlights how it outperforms other state-of-the-art protein docking methods. HDoc was ranked as the top performing docking server for multimeric protein structure prediction, as can be seen in figure 5, A through C. HDoc had a 100% success rate for easy targets, a 27.3 success rate for difficult targets, and a 60% overall success rate, which is about 12 out of 20 targets overall. The consistently high performance across all target categories emphasizes the robustness of the HDoc software. Figure 5D further demonstrates HDoc's strength by showcasing its top-ranking performance among the 38 CASP13 Capri groups, specifically for cryo-EM, which is a particularly challenging class of structural prediction. This further solidifies HDoc's capability to handle even the most complex structural data and outperform competitors like HawkDoc in accuracy and across a range of docking difficulty levels. Now, tables 6 and 7 demonstrate how the HDoc server performs quality checks on input structures before performing docking. This helps the users assess the reliability of their docking results without having to run quality checks manually. Table 6 is the quality criteria of user-provided input protein structures by ProQ. The ProQ program evaluates quality based on two scoring metrics, LG score and max sub. LG score measures overall quality of the structure by comparing it to known native structures, and typically ranges from 0 to 15, and the higher the LG score, the better the model is. Max sub measures structural similarity between a model and a native structure and focuses on the alignment of well-modeled regions. It typically ranges from 0, meaning no match, to 1, meaning perfect match. And if both the LG score and the max sub fall below the threshold for even the correct level, the system issues a warning saying the quality of user input protein structure is low. Now, table 7 is the quality criteria of homology modeled protein structures from amino acid sequences by TM Align. HDoc generates models using homology modeling and then assesses their quality with TM Align. The TM Align program evaluates quality using two scoring metrics, seq ID and TM score. Seq ID measures the percentage of amino acid residues that are identical between the input sequence and a known template. It ranges from 0 to 100%, and the higher percentage means a higher quality homology model. TM score measures the structural similarity between two protein structures and is normalized for protein length. It ranges from 0 to 1. The higher the number, the higher the confidence is that the model is structurally accurate. TM score is similar to root mean square deviation, but it's preferred because it's less sensitive to local errors. Like ProQ, if both the seq ID and the TM score indicate low quality, the system will again issue a warning. The quality of the homology model is low. These tables reflect HDoc's commitment to transparency and user guidance. By automatically screening input quality and clearly reporting it, users can interpret docking outcomes more accurately, decide whether to trust the results or refine their input data, and also to avoid wasting time analyzing unreliable methods.
To summarize, the purpose of HDoc is to be able to predict protein-protein interactions through the combination of ab initio template-free docking and template-based modeling. The input for HDoc is amino acid sequences, PDB structures, or even both. However, these inputs do come with limitations, one of which is structural accuracy. If you provide a PDB structure, its quality is determined by the technique that was used to determine the structure. For instance, this could be X-ray crystallography or cryo-EM. Certain techniques provide worse resolution than others, which decreases structural accuracy. If you provide a sequence, you can also potentially get an inaccurate structure. This is because the current HDoc server can only model single-chain proteins from sequences. Therefore, if the protein of interest is biologically active as an oligomer, HDoc will not be able to model it unless the user is also able to upload a 3D structure as well. Another limitation is molecular flexibility. The HDoc server uses a rigid body docking approach. This means that the receptor and ligand are treated as rigid structures when they are mapped onto grids. However, the ligand or receptor can experience large conformational changes after binding. While the grids can assume some level of flexibility, they don't assume it to this extent, which can lead to errors in docking results. This limitation can be greatly reduced by having a homologous template available for both the ligand and receptor. This is because template-based docking accounts for conformational changes to a much greater extent than template-free docking. There are some assumptions that we have to make about the inputs as well. One assumption users of HDoc make is that the structures they input are biologically relevant. In other words, they have conformations that would be active in the human body. However, this might not always be the case, especially if you use a crystallography technique where the inactive conformation is crystallized. This can result in major docking errors. Another assumption is that the interface of the two proteins can be reached with the starting conformations that are uploaded. Once again, as mentioned before, the proteins could be in their inactive forms in which the active site on the receptor molecule is not accessible to the ligand molecule. In reality, these molecules would not reach an interface because they wouldn't be able to bind together. However, the HDoc server would still suggest a different interface based on the inactive conformations, which leads to an incorrect prediction. The output that HDoc provides us with is potential 3D structures of the protein-protein complex that we are interested in. This includes the top 100 docking models as well as the top 10 models for visualization, but just like the inputs, the outputs have limitations as well. One limitation is the accuracy of protein homology models. 3D structures generated using homology become less accurate with less sequence identity between the target and template. Low sequence identity can lead to many errors in the structure, which results in an unreliable model. Additionally, the HDoc server is able to predict 3D structure for easy and medium difficult targets. However, it sometimes cannot find a template for more difficult targets, so the results page gives no structure predictions. Another limitation is complex accuracy. As mentioned earlier, the server assumes the receptor and ligand are both rigid structures, so HDoc doesn't model major conformational changes. If binding of the two components requires significant movement or folding, the predicted model may not show it, indicating that the results can be unreliable. Additionally, the predicted model may not be biologically accurate. As said before, if the inactive conformations of the proteins are the ones that are docked at the very beginning of the process, the results would show a completely different interface than what happens in the human body. And then there are also assumptions that come with the output. The models generated are based on the assumption that the proteins are pretty rigid, meaning they don't undergo large conformational changes. And this assumption is made because the HDoc server assumes at the very beginning that both the receptor and ligand are rigid structures. And there is also the assumption that the input structures are in their active native forms. Scientists using HDoc are searching for the structure of the functional version of a protein complex they're interested in. So they assume that the structures they put into HDoc are actually able to bind together to create that complex. Considering the size, oligomeric nature, and catalytic function of beta-galactosidase, could the HDoc server still be used to study that protein? Yes, it can. Despite beta-galactosidase being a large complex tetramer, the server can be used to study the activity of one of the protein's monomers, and that can be done in multiple ways. First, users can study the monomer-monomer interface in order to explore the formation of a functional tetramer. They can also study how a single monomer interacts with different activators and inhibitors, and this can give insight into how regulatory molecules affect enzymatic activity. Mutations can also affect enzymatic activity, and they can occur within a monomer of beta-galactosidase or within the monomer-monomer interface. And studying this can help scientists predict how mutations affect the overall stability of beta-galactosidase and the strength of its interfaces. The data generated from HDoc can be used in different ways. To begin with, HDoc provides us with the top 100 docking models for our complex of interest, as well as the top 10 visualization models. And these models can be used to help us narrow down potential binding sites or even some interface residues. However, is the HDoc server biologically relevant on its own? Not necessarily. While it does provide a good starting point and allows users to form a testable biological hypothesis, it only provides us with predictive structures for a protein-protein complex, not definitive ones. However, this makes HDoc good as input for other tools, such as biochemical assays that can confirm biological relevance. One example of which is enzymatic assays that test functionality, and another example is site-directed mutagenesis that tests the importance of predicted interface residues.
So to conclude, HDoc is a great example of how computational tools are becoming more accessible and more powerful at the same time. It allows researchers, even those without structural biology expertise, to predict complex interactions between proteins or nucleic acids using just sequences or known structures. Its hybrid approach is what sets it apart, honestly, by combining template-based modeling with template-free docking. It balances speed, flexibility, accuracy. The results from benchmarking studies show that HDoc consistently outperforms many other tools. Obviously, no tool is perfect. HDoc's rigid body assumptions mean it may miss key structural flexibility, and predictions from sequence alone can vary in quality depending on available templates. That's why experimental follow-up is still essential, especially for more biologically complex systems. But with rapid development and the possibility of integrating flexible docking and as well as AI-based modeling, tools such as this may soon push us closer to answering bigger biological questions faster. To end with, our group came up with the question: as these predictive tools continue to evolve, should experimental biology start relying more on these computational predictions, or will hands on validation always be essential? And here's our works cited.